What's happening on Friday? Midterm, yes. How many clicker questions today? I guess the word got out, given the number of the people who are here today. So hopefully it will be useful to you to be here today. We shall see. As always, ask questions, slow me down, more questions come up, grab me in office hours. If people really feel the need, we can try and set up some extra office hours as well before the exam. So let me know. May have some time afterwards to do that. We shall see. I did actually finally get around to adding a couple of extra slides to the presentation from yesterday, put them on YouTube. I don't know how many people noticed. It's kind of a funky audio-video mixture, but hopefully that's all getting taken care of, and I'm trying to record this one as well. But I put in all the extra slides that had somehow gotten cut off last time. The new version of the software, at least in theory, makes that possible. So again, I'm going to go through stuff really quickly today. This is things that I think usually I've done a pretty crummy job of explaining, mostly because people have asked me more questions about them later on. And then I actually noticed in one particular place that I actually made a mistake on one of the slides. So wanted to go through that. But in general, we're going to go over this stuff really pretty quickly. Way back when, yeah, feels like forever ago, we talked about the histone code, and particularly histone modifications. And just here wanted to emphasize that you can have lots and lots of different modifications, actually not just on the N-terminal tail of histone H3, histone H4, histone H2A, H2B. You can also have modifications that happen inside the core part of the histones here as well. These modifications, for instance here, position lysine 23, are actually mutually exclusive. So you can either acetylate or you can methylate, but you can't do both at the same position at the same time. But you can have acetylation and methylation happening at the same time, just on different side chains. And what these mean, quote unquote, and I love the fact they use quotes here as well, is that these modifications can mean lots of different things. Usually trimethylation, and particularly trimethylation at lysine 9, leads to gene silencing and heterochromatin formation. On the other hand, trimethylation at lysine 4 in combination with acetylation at lysine 9, that leads to gene expression. So it really kind of bounces all over the place, and that's kind of why the code and the meaning here is really in quotes, because it's pretty variable. So I'm not going to expect you to remember all of these things because scientists don't even completely know exactly what all of these things are. But that you can't have modifications at the same place at the same time, and that methylation and acetylation can mean multiple different things depending on the context of where they are. How these modifications are noticed, last question on last exam that evidently I didn't do a good job of explaining because many people missed. The recognition of these modified histones happens through the interactions with other proteins. And that's particularly here, the code reader proteins. So histone code modification, which is a little blue dot, that gets recognized by a protein-protein interaction. In many cases, this reader protein also is connected to a writer protein. And so this way you get the positive feedback loop. If you have one modification of one histone, it's likely to cause a modification of a histone right next to it in the same way. Always end up with this chicken and egg question. How do you get those modifications in the first place? That's usually due to gene regulatory proteins that we'll be talking about after the midterm, which will bind specifically to DNA in one position and then start off this whole chain by interacting with one of these writer proteins. Then, of course, if you've got these feedback loops, you clearly have a problem to stop this kind of positive feedback. So then you need barrier proteins. These can be structural, they can be enzymatic, and those will block the progression of this process. The other thing about these positive feedback loops is that once you have replication, the modifications that have already been made in that part of the genome that's getting replicated, those can be inherited because some of the histones that were there will reassociate with the DNA after it's been replicated, and then lead to this kind of positive feedback loop in the process. That was histone modification. Again, one of these things we could spend a whole course talking about, and we'll talk more about it when we talk about gene regulation later on. When we think about chromosomes, most people usually think about mitotic chromosomes down here. Mitotic chromosomes are incredibly compacted, and in such a way, again, that you go from the 2 odd meters that we would have of DNA if it were in B form in every single one of your cells to, well, it actually can fit into a nucleus, and particularly it fits into a nucleus so you can undergo metaphase. So this completely compacted form is way too compacted to have any kind of machinery associated with it. So mitotic chromosomes are not active for transcription. They're not active for replication. They've only been compacted in such a way that they can actually end up being separated. The place in compaction where you can actually get transcription take place is here, where it's a extended form and mostly in this 30 nanometer chromatin fiber. This is interactions of the various nucleosomes with each other, and then a lot of other nucleosome binding proteins. So this is the state that probably most of your chromosome in interphase is present in, and where you're actually getting transcription taking place. This needs to be compacted and literally condensed through the activity of those condensins that we talked about. So the condensins grab the loops of DNA and then stack all of those loops on top of each other in order to give you a complete mitotic chromosome. A couple of people asked me about puffs. What are these puffs? What's a chromosome puff? Well, chromosome puffs happen in a very specific kind of cell under very specific kinds of conditions. And so this is actually not what our chromosomes look like at any particular time. This is in salivary glands of some insects. You have lots of interphase chromosomes all lined up on top of each other. So it's many, many copies of the chromosomes that have not divided. But they're still in interphase, they're still active. And the reason for this is that a lot of these salivary glands, they need to make a whole bunch of some proteins. And so they need many copies of those genes to be there. When they're all lined up next to each other, you have this pattern of bands, dark bands, light bands, dark bands, light bands. That is for the majority of all of these chromosomes that are lined up next to each other. When genes are being expressed, i.e. they're being transcribed, some of these bands will disappear. And that disappearing is they become decondensed. And that decondensation is where you now have all of the RNA synthesis taking place. So the puff in a chromosome is where decondensation has happened so you can have all of the transcription machinery. And as we've talked about probably ad nauseum now, it's not just the RNA polymerase, it's all the proteins that are involved in getting the RNA polymerase to the promoter. It's all the proteins that are involved in splicing, in tailing, in capping, etc. So really the mRNA factory that has to be associated here. And so all that these puffs are is just a way of visualizing in fact, a light microscope. You can see these bands, and if a band disappears, that means, oh, it's become decondensed, and it's probably going to be being transcribed. So it's just a nice way of visualizing things. Another way of visualizing this decompaction and also the movement of wherever your genes are that are being transcribed to a separate place here in the nucleus of a regular cell. We have where the chromosome is, and these are the 2 homologous chromosomes, different parts of the nucleus here in green, and the little pink dot is where you're going to have an active gene. Here that gene is not active, here the gene is active. It's clearly moved to somewhere else in the nucleus, and that's where your transcription factory, your messenger RNA factory happens to be. So any questions on chromosome structure before we move on to replication? Okay, so replication, yes, semi-conservative, etc. The main confusion as far as replication is concerned has to do with the experiment that Mismatch and Stahl did and how they figured out what was going on here. The real critical thing about measles and Stahl's experiment, and true for all DNA, it's kind of hard to take two strands apart. That's why you need helicases. That's why you need the huge amount of ATP hydrolysis. So when Muslin and Stahl were isolating DNA that was made by the E. coli, all they could see was double-stranded DNA. So starts out double-stranded. They isolated double-stranded DNA. After the first generation, they isolated double-stranded DNA after the second generation and double-stranded DNA after the third generation. We now know that these are hybrids between new DNA and old DNA. But you've got to be able to separate the double strand. So here's double strand both old, double strand old and new, so it's half, and then the continuing halves and completely news. And so that was what their experiment was about, and that's where they did the equilibrium ultracentrifugation, separating the DNA double strands now based on their density. And what they saw was you start out with one density here. This is the heavy nucleotides. And then after 0.3 generations, they purify the DNA. It's actually mostly still the DNA that they started with, but they're starting to see some DNA which is a little bit lighter, and so on and so forth. After one generation, it's this intermediate form, which is clearly not the same as they had here, so that rules out any kind of conservative replication. And then as you get to the second generation or near to the second generation, now you start to see yet another separate. So these are now double strands. They're double strands which are just light. So it's separating just heavy, heavy and light, and just light from each other. And that's the key to this density experiment and how they had it done. So is that clear to people? Were someone to give you a question on an exam about the other kinds of models, you would know what they would look like? Yes, good. Someone to give you something like this, you could figure out what generation it was? Yeah.
What would it look like if it was conservative replication?
If you had conservative replication. So if you had conservative replication, let's back up. So you would end up with new, and old as double strands. And you just end up with more of the new. So you'd end up with two bands. Sorry, wrong direction here. You'd end up with two bands, one here where you'd started with, and one up here which is new. And you'd never see an intermediate band because you'd never have half and half. That make sense? Yeah. Okay. What are questions on the most beautiful experiment in biology? People would argue about that, but that was what Gunter Stent said, and don't argue with Gunter Stent. Well, you can't. He's died a couple of years ago. But so they wanted to mention the DNA polymerase. A lot of questions have come up, and which is a reminder, if you have a question you'd like to ask me, please check D2L first under the discussion and questions linkages, because I've actually answered quite a few questions there already. Some of those have to do with DNA polymerase 1 versus DNA polymerase 3. Why the heck is it called DNA polymerase 1 when the replicative polymerase in E. coli is DNA polymerase 3? The reason for that, it was the first one that was found. And the reason it's the first one that was found is the one that was the most abundant in cells. And we'll see why that is, of course, in just a second, because it's important on lagging strands and dealing with all the primers. If you think about DNA polymerase 3, the replicative polymerase, there's only potentially 4 copies of that protein per E. coli cell. So there are very, very few copies of that particular protein, which is why they found DNA polymerase 1 first. Other things about DNA polymerase 1, and actually all of the DNA polymerases, and for that matter, the RNA polymerases, is they all go from 5' to 3' because they're adding a 5' triphosphate to a 3' OH. And that's because if you tried to go the other way and tried to do proofreading, which all of these enzymes, DNA polymerase 1, DNA polymerase 3 do, you couldn't add any more nucleotides. And so going in this direction, losing your pyrophosphate in this polymerization process allows you to have this proofreading activity. And the big problem, and apparently Arthur Kornberg used to bug his students and postdocs about this as well, is that it also had this strange exonuclease activity that they couldn't get rid of. So they had the proofreading activity, which is the backing up. That's the 3' to 5', but it also had 5' to 3' exonuclease activity. And that was really bizarre because it has polymerization activity and also this exonuclease activity. It's kind of spinning its wheels. It's really not ending up giving any kind of replication. And that's why they went back and looked for other polymerases, is they wanted to find a polymerase that didn't have that kind of exonuclease activity, so it could be the replicative polymerase. Sorry, I missed your question.
So you're saying it proofreads in both directions?
Okay, no, so proofreading is by definition the backing up. So they have two exonuclease activities, and that's just the enzymatic activity. The enzymatic activity can be 5' to 3' or 3' to 5' in the case of polymerase 1. The proofreading activity is the backing up. So 5' to 3' polymerase, 3' to 5' exonuclease, that's your proofreading activity. But yeah, the 5' to 3', as we all now know, is for getting rid of primers together with RNase H. And that's why, since you need to get rid of primers, you've got a lot more of DNA polymerase 1 than you have of DNA polymerase 3. A lot of confusion came up with this table, finally figured it out later, having to do with this fidelity. Fidelity, i.e. how often the wrong nucleotide gets incorporated. The most fidelity actually comes from the polymerization. And this polymerization step is just the polymerase checking to make sure that the geometry of the base pair is correct. It has nothing to do with the identity of the bases. It's just that you have a purine pyrimidine that are matching together with each other. If you have proofreading, which is this exonucleolytic backing up, that will correct about 1 in 100 of the misincorporated bases that you ended up with from here. And then this is the confusing number. Here in our textbook, it's 10 to the 3. In all my slides, it's 10 to the 2. I went and looked it up. It varies from 1 in 50 to 1 in 1,000. So both of them seem to be right, myself and the textbook. So if there were a question about something like this on an exam, how often does strand-directed mismatch repair 1 in 1,000 or 1 in 100? The answer is yes.
So this table is actually showing how frequently the repairs are made, not how frequently the errors are made.
No, so this is how frequently you end up with an error. So after polymerization, you'll still have 1 in 10 to the 5. After proofreading, you'll have an additional 1 in 10 to the 2. And after mismatch repair, it'll be 1 in 10 to the 3 or 1 in 10 to the 2, depending on which system you're looking at. That will give you 1 in 10 billion or 1 in a billion, which is the other number which I keep using, which is the 1 in a billion. So it depends on how frequently this strand-directed mismatch repair is happening. Yeah?
You want us to memorize all the numbers, right?
So I would say know the numbers, but know that this number, the 10 to the 3, could also be 10 to the 2. So if I give you a multiple choice answer like this, it should be one or other of those two. Okay, and I know I've not finished with the exam yet. I've got 48 hours too, just like you do. So that being said, one thing that I think I did a really crummy job of explaining the first time around was talking about replication forks. And so the idea here is once you have the replication origin, you have starting of replication, what you end up with is 2 forks, literally, which you can kind of see here. Here's the DNA that hasn't been replicated yet. Here there are DNAs that have been replicated. And literally right at that junction is what's called the replication fork. Whenever you have an origin of replication, at least that we've talked about in this class, you end up with 2 replication forks. So replication fork going this way from an origin and a replication fork going that way from a replication origin. Each of these will have a lagging strand and a leading strand. The leading strand will be the one which is being synthesized continuously from 5' to 3', and then a lagging strand will be made discontinuously. So it's a discontinuous semi-conservative, semi-discontinuous because this one is continuous, semi-discontinuous, semi-conservative replication. And then these Okazaki fragments that have to be made with primers. And again, that's where you need the DNA polymerase I to get rid of some of these primers. Another thing that I forgot to mention is that bacteria and archaea too are infinitely superior to eukaryotes in many different ways. But one of them is that the size of Okazaki fragments in bacteria and archaea is pretty big. It's about 1,000 nucleotides. It's a lot shorter in eukaryotes. And that probably helps with mismatch repair. Because you remember mismatch repair has to do with where you have nicks in your DNA on the newly made DNA, which is, again, in the case of eukaryotes, about every 100 nucleotides or so. Yeah? Oh, okay. So the question, sorry if I'm misunderstanding you here, but the question is really about how long those Okazaki fragments are. So it's not the replication fork per se. The replication fork will continue and go all the way around the genome. But it's the Okazaki fragments and how long they are before it stops and restarts. And so in bacteria, it turns out that's about 1,000 nucleotides. And in eukaryotes, 100, 200. Mm-hmm.
Why is it that low? Why are those replication forks placed
Where
where they are?
or maybe better question is why the origins are placed where they are. Because where replication forks are is going to depend on where your origins are. Okay. And we haven't really talked about that, but a lot of that is because it's not entirely clear where origins are. And that's defined by where that origin recognition complex is going to sit. So we'll talk about that in just a second. But it doesn't seem to be as specific. So for E. coli, we talked about OREC, the origin of replication for the chromosomal replication. That's got binding sites in it, very specific DNA sequences that DNA A will bind to. And once that happens, then you get the origin that happens right there. Origins in eukaryotic systems, and eukaryotic systems have really been a really great word for this because it turns out in some eukaryotic systems like yeast, the origin is pretty well defined. In human chromosomes, it's way less well defined. And so where you have an origin of replication seems to be pretty variable. It doesn't even always happen in the same place at any given chromosome in any given place where it's replicating. So yeah, it's a much more inefficient process because another thing that I didn't mention, but the polymerases are also a lot slower, the eukaryotic polymerases, probably because they're having to deal with nucleosomes and all of these other things as well. So polymerases are slower. You've got bigger genomes mostly to deal with, and all of these extra replication forks that are banging into each other. So way more complicated than the nice, simple, straightforward, streamlined bacterial process. Does that more or less explain? OK, thanks. So one thing that you would clearly then have to have more of in bacteria, because you're going 1,000 nucleotides before you restart each of your Okazaki fragments, you have to have single-stranded binding protein, because till that polymerase gets done with the Okazaki fragment, that's going to jump back and start again, because it's the opposite orientation on that lagging strand. You need single-stranded DNA binding proteins. And single-stranded DNA binding proteins will protect that DNA from getting chopped up, but probably at least as important is stopping the DNA from base pairing to itself, forming these so-called secondary structures where the base pairs form on a single strand of DNA. DNA polymerases have a lot of trouble with these kinds of base pairing interactions. So single-stranded DNA binding protein stops that, and it also protects this single-stranded DNA from getting degraded. Another thing that I forgot to mention this highly cooperative. What do I mean by cooperative binding? We'll talk more about cooperative binding when we talk about DNA binding proteins, regulatory proteins later. But the basic idea here is once you have binding of one protein, then it will stimulate the binding of a whole lot more proteins. So that first step of binding has to happen, and that's a relatively slow step. But as soon as that first one is bound, then you have a binding of a whole bunch more. And so basically, as soon as you make a single-stranded piece of DNA, these single-stranded DNA binding proteins will coat it very, very quickly. Same thing is true with Rec-A in terms of getting homologous recombination binding to those single-stranded pieces. Another thing I wanted to mention, again, I think I did a crummy job of explaining before, processivity. Processivity of DNA polymerases. DNA polymerases by themselves, in the absence of extra factors, are not processive, i.e. they fall off of their template all the time. And so that's what's supposed to be shown here on this side, non-processive DNA polymerases. And this non-processive DNA polymerase would add one or a few nucleotides and then fall off the DNA. This is a problem because if you fall off all the time, then you've got to come back onto the DNA and bind it every single time. On the other hand, if you had a processive DNA polymerase, and DNA polymerase by itself, again, is not processive, then it will stay on the DNA and add lots and lots of nucleotides. And it turns out that this was studied relatively early on in the study of DNA replication by just taking a DNA replication reaction and diluting it, just putting in a whole bunch more buffer, and then seeing how fast replication was after you'd done that. If the DNA polymerase were falling off of the DNA all the time, then that should have slowed down your DNA replication really majorly, bigly. No. So what happened, however, was you do that dilution, and DNA replication continues going on at the same rate. And so what that means is there's something else, which of course we now know is the sliding clamp, which is holding the DNA polymerase onto the DNA to make sure that it doesn't fall off until it gets to a termination point. So that gets us to our replication fork. And again, this is just one of them for any particular origin. Every origin, you're right, it's going to go in both directions. So whenever you have an origin of replication, you can have two replication forks, two leading strands, two lagging strands. These will have DNA polymerases associated with them. Then the leading strand also has to have a sliding clamp. This is synthesized continuously. The lagging strand also has this polymerase attached to it, and with a sliding clamp also attached to, not shown in this cartoon, the leading and lagging strand polymerases are attached to each other, and all of them together in the replication fork. DNA primase will be putting down primers. Once you have this RNA primer that's made, that's the clamp loader process. This will generate lots of single-stranded DNA through the activity of your DNA helicase. This now will be replicated from this primer, getting rid of this single-stranded DNA priming, single-stranded DNA binding protein to get to an RNA primer. At this point, now the replicate DNA polymerase actually falls off of the template. It's still attached here through protein-protein interactions, so it just goes back and finds the next primer. This is where you have DNA polymerase 1 in E. coli and the FEN1 protein in eukaryotic cells. It gets rid of this primer. You also have RNase H. There are two mechanisms that are being used for getting rid of primers: RNase H and Pol1 in bacteria, RNase H and FEN1 in eukaryotes. So, question of replication, yeah?
I remember one time you mentioned that DNA primase uses ATP. Can you explain why?
And explain why DNA primase uses ATP.
Or
why or else. So, you could say if you happen to have a primer which didn't have as a template any T residues, then it wouldn't be using ATP because it would be using CTP, GTP, and excuse me, was it CT and the other one be UTP, actually, because it's a RNA polymerase. So that RNA polymerase is also going to use ATP, but ATP as one of the nucleotides is going to be adding. So in the primer, it's using ATP, but using ATP as one of the nucleotides is adding as the primer. So it's not an ATP hydrolysis for energy per se, it's ATP as a substrate for polymerization. Yeah?
Why use RNA primer as opposed to DNA primer?
Why use RNA primer instead of a DNA primer? It doesn't need a 3' OH. It doesn't need a primer. DNA polymerases all need primers. And so you can't start. DNA polymerases by themselves cannot start without an RNA to start from, or a 3' OH. Take virology next term. You can sign up for it now. You'll hear about all the other cool ways that viruses have figured out how to do this without RNA primers. But as far as we're concerned, the RNA polymerases can start. Those will start from wherever and generate then the 3' OH that these DNA polymerases can then use. Yeah?
On the last slide, you were talking about the processivity.
Mm-hmm.
Is that driven by the sliding clamp?
Okay, so the question is processivity driven by the sliding clamp? I would say change that around a little bit and say processivity is allowed by the sliding clamp. And so it turns out you also have DNA polymerases that you don't want to be processive. And I didn't put these in the slides. Which ones? Error-prone polymerases, the translesion polymerases. You don't want those to be processive. And so those are not going to interact with the sliding clamp and stay that processivity. So yeah, that's, you'll want to have some that do and some that don't. Okay, what happens when you pull apart two strands of DNA? You end up with superhelical problems, and I brought my extra models here so you can come play with supercoils, since I don't sure I even have a phone with a cord anymore. But what happens here, as soon as you pull two strands apart, literally is pulling these two strands apart is untwisting them. Because if you pull them apart, they're not wrapped around each other anymore. That untwisting has to be compensated for somehow, and the way it gets compensated is through these supercoils, which are generated, also known as writhe. Either kind of topoisomerase can deal with these. Topoisomerase 2, which cuts both strands, requires ATP, or a topoisomerase 1 that just literally cuts one strand, allows it to religate on the other side. That does not require ATP. It's a OH on your tyrosine that just gets exchanged for the OH in the 3' and is a reversible reaction. Pretty clear on topoisomerases. Again, could spend a whole course talking about them. Other thing that I wanted to talk about, re-mention, and this actually ties in really nicely to DNA replication. Big problem with DNA replication if you have a linear chromosome is what do you do with the ends, because you need a primer, right? It's only the RNA polymerases that can make primers. You don't want to keep those primers around, because we didn't talk about the fidelity of RNA polymerases is way lower than the fidelity you have in DNA polymerases. So RNase H, DNA polymerase 1, the flap endonuclease get rid of these primers. But if you get rid of a primer at the very end of a genome, now you don't have a template anymore to make that opposite strand. So what do telomerases do? Telomerases make templates. And so the telomerase is a reverse transcriptase. Reverse transcriptases are also DNA polymerases. So for the DNA polymerase, it has to have a primer. So what's the primer here? The primer is actually the 3' end of what was already made by all the other DNA polymerases. This was the primer that was removed on the other side here, because that was the RNA that was used to make this piece. So here, 3' OH, that gets extended by the telomerase. So extended, 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 extended. Now you've got a really long template and enough space for a new RNA polymerase. The DNA primase sit down make a short RNA that'll get extended by your DNA polymerase and literally fills in this template which was made by the telomere. Once it gets to the end, there's still going to be this overlap because that second strand is all being made by regular polymerization machinery, DNA primase that makes RNAs, and the DNA polymerase that comes in after that. So you're always going to have this little end that's hanging out here. This end can base pair to the sequences which are over here because they're repeated sequences. That's how you form this T-loop. So that single-stranded end here loops around and will base pair right here and actually bumps this strand out of the way where that base pair is formed. And this is the T-loop structure. This protects the end of the chromosome from getting chewed up by all the hungry exonucleases that are inside the cell. Also, there's a lot of other proteins that are associated with it that we're not going to talk about here that also says, hey, this is not a double-stranded break and don't do recombination here. So that's what happens at the ends of the genomes. How do you start? Kind of getting back to your question. Origins of replication. This is where I made a mistake, a bad one, so that's why I've corrected it here. DNA-C is actually not the clamp loader. It's the helicase loader. DNA-A binds to replication origins. It's the initiator protein. And after it binds to these origins of replication, it brings in other proteins. And so this whole process, first you have DNA binding by a specific DNA binding protein, and then you bring in other proteins that associate with this. This seems to be what happens in molecular biology all the time. Specific binding of a DNA binding protein, and then protein-protein interactions bringing in many more proteins. So the real key to this process, and really for all replication processes, is pulling apart the two strands of DNA. And how do you pull apart the strands of DNA? It's that helicase. So as soon as the helicase is activated, then you have activity of the primase. Primase is going to give you a 3'OH. The DNA polymerase will start to make their leading strand. As soon as you have enough of this lagging strand here, there'll be enough for that DNA polymerase to start making. And you have two replication forks, one of which will go off here to the left, and one of which will go off here to the right. This binding again to origins, that's where you get everything that starts. It's different in eukaryotes in that you have a different set of proteins. Yeah?
So DNA-B and C are helicase?
No. So DNA-C, sorry, is a helicase loader. So I just crossed off the clamp. I didn't cross off the loader. So you need all of these proteins at the origin of replication in order to get your helicase to be functional. And once it's functional, then you get the rest of replication to happen.
So how does helicase go on before the loader then?
No, it's just, these are just the different names. The order here is you have helicase and, in this case, it's DNA-B and DNA-C together, which bind to DNA-A through protein-protein interactions, and then you have the helicase associated with your DNA strand. So it's not that it happens in this order here. Now, the order is what happens here in the eukaryotic system. You have the origin recognition complex. And another question that's come up is origin recognition complex. One of the things that you will hopefully notice here is this origin recognition complex is bound the whole time to this origin of replication. So before you have replication, after you have replication, it's always bound to this particular position. And this is the one, getting back to your question, where is it binding? And where do you have that origin recognition complex bound to DNA? And in some cases, and actually turns out in yeast, you do have some pretty specific sequences that it binds to. In larger eukaryotes like us, it seems to be kind of all over the place. And so there's not a real nice, easy way of saying where it's actually going to start. But if you have the origin recognition complex and you're going through the cell cycle, you have association with these cell cycle proteins, CDC6 and CDT1. These then bring in the replicative helicase. That replicative helicase actually just sits here all of these complexes until the cyclin-dependent kinases are turned on, and you're just starting S phase. And this process, the G1 to S transition, there's all kinds of really cool regulatory mechanisms that if you're taking cell biology, you probably heard way too much about, and all the proteins that are involved in that process. But as far as we're concerned, cyclin-dependent kinases are turned on. That will lead to dissociation of CDC6 and CDT1, which allows the helicase to pull apart the two strands. And just like the case that we have in bacteria, as soon as those two strands are pulled apart, then you have DNA primase, you have all the DNA polymerases, sliding clamps, et cetera, which you can get for replication forks. So it's getting that helicase activity, pulling the two strands apart, which are the key to getting replication started. So again, activity of helicase, which is the most important. So that's it for replication. We good with replication? Yes? No? Okay, DNA damage. DNA damage, again, a couple people have asked me about this. This is probably the most important in thinking about the different kinds of DNA damage here. But the size of the arrows here is what we really care about. So big arrow right here. Hydrolysis of the bond between the base and the sugar happens most frequently with purines, so depurination. Here, big blue arrow, deamination of cytosine. These are the most common kinds of DNA damage that happen inside of cells. Common DNA damage, this is going to get repaired by the base excision repair machinery because they're very common, they're very specific. You also will have some other ones, and in fact the large arrow here, somebody asked me about this before, oxidative damage, where does that happen? Here at this 8 position in the guanine, 8-oxo-G, quite common, also has a specific DNA glycosylase that will deal with that, take that very specific nucleotide out that's been modified. I wanted to also mention this methylation that happens here at N7. This should sound really familiar because that's exactly the same position where that G gets methylated in cap structures. So 7-methylguanosine cap is the same place that you can also have 7-methylguanosine. However, 7-methylguanosine in DNA is bad and it gets chopped out. How do these things get dealt with? We talk about base excision repair, but mismatch repair, this again is where we have some confusion. Here 10 to the minus 2, in that table it was 10 to the minus 3. So these need to cut out the wrong nucleotide once it's been added. And so how does the cell know that it's a newly added nucleotide? Bacteria, it's easy because it's the non-methylated strand. So non-methylated strand, that's the newly added one. Anything that's the newly added one, which is incorrect, you need to get rid of. On the other hand, eukaryotes, because they've got these short Okazaki fragments, have a way of figuring this out. They just say, we've got your nicks. So when you haven't gotten rid of your primer, you haven't actually completely filled in those Okazaki fragments, it's really easy to figure out where you have that mismatch that's taken place. Mismatches are detected by a problem with the structure. It's the wrong base pair. These things are not interacting with each other. There are proteins that will specifically recognize that, and then make nicks. In the case of bacteria, 2 nicks, one on either side. And a nick is just cutting one of the strands. Cutting one of the strands between the phosphate and the base. Once that's been done, then you have helicase activity, gets rid of it. That helicase activity now has a single-stranded template, a 3' OH, and this can get repaired. Yeah, back.
If the methylation occurs, what process will happen? Because then what, like the mismatch, not be detected?
Okay, so methylation in terms of DNA damage, or methylation in terms of how the cell, in this case the bacterial cell, knows what's old DNA versus new DNA? Okay, so how does it know between old DNA and new DNA?
Yeah.
Okay, so when does that methylation happen, maybe? Is it maybe a better question? So I'm not quite sure I understand your question, though.
So like if a methylation damage occurs,
Yeah.
how does
Okay, so these are different methylations. And so the methylation that happens here is actually at a specific sequence. Here, CTAG, and it's this A position right here, which gets methylated. That is not recognized as DNA damage. And it's because there's very rare that you have methylation that happens there through a damage process. On the other hand, 7-methylguanine, which is the one I mentioned as being the really common one, that then is recognized by a very specific enzyme that says, hey, is there a 7-methyl-G here? Then I will take that out. Yeah?
You referred to the nick as being between the phosphate and the base?
No, sorry, between the phosphodiester backbone. I'm sorry if I made a mistake on that. Yeah, no, it's not getting rid of the base. Yeah?
I was wondering, how does this cause gene conversion?
Okay, we'll get to that in just a second. Yeah.
What's
HNPCC, hereditary non-polyopsis colon cancer. It's a particular gene which is mutated in these hereditary colon cancers, which is involved in mismatch repair. So that's why I have it up here. And we also when we talked about repair, a lot of the problems in DNA repair mechanisms are also mutated in lots of cancers. Okay, so maybe this will help you with your question in the back here as well. So for the very common kinds of DNA damage, there are very specific glycosylases which do cut the base off from the ribose if there's an incorrect one. And the classic example is uracil, which is deaminated cytosine, but also for 8-oxo-G or the 7-methyl-G, something like that. These very common ones will have a specific enzyme that will take that damaged nucleotide out of the DNA. And so once it's done that, now you have a P site, a pyrimidinic, a purinic site. This is what happens with the most common form of DNA damage, which is depurination, just due to heat. And then this single nucleotide gets cut out, it gets repaired. This is a very efficient mechanism because all you're doing is changing one particular nucleotide, but you have to have the enzyme up here which recognizes that very specific kind of change. And there's lots of other kinds of DNA damage that can happen as well. Or if you happen to have a mismatch, that's not the wrong nucleotide. It's actually a correct nucleotide. This is a correct nucleotide in the wrong place. So you can't use base excision repair to get rid of correct nucleotides. You can only use it for these damaged nucleotides. But what happens there, in terms of a mismatch, which also happens with a lot of other DNA damage, is that the structure of the DNA is wrong. You don't have the right B-form DNA. And so that's where you cut on either side and get rid of that piece and resynthesize it. Okay, so we're getting to mismatch the gene conversion here in just a second. So that's great if you have problems on one strand. If you cut both strands at the same time, then you can't use the opposite strand as your backup. You can't use that as the template to repair the problem that you had in the first place. And so the way this is done is through double-stranded break repair, and it's not just for repair. It's also for meiotic recombination. So meiotic recombination, you have binding by and cutting of your DNA by these proteins, which gives you a double-stranded break. Then you have a exonuclease that chews in the 5' end. And then we have a single-stranded piece, which will be homologous to a different chromosome. So in bacteria, this is usually where you just have a replication fork, so newly replicated DNA. In eukaryotic cells, in particular in terms of meiosis, you have homologous chromosomes that are going to be used to have this repair mechanism. So once you have this 3' end, that gets bound to Rec A in bacteria, various rad proteins in eukaryotes. Then you have strand invasion. And all that strand invasion is it takes that single strand, pushes the other strand away, and now provides a 3' OH. And you can have DNA synthesis that takes place from this position. That DNA synthesis continues here. this displaced strand will also match the other 3' extension from over here. That will also provide DNA synthesis. Once it gets to this end here, these guys can get ligated together. This one will also get ligated over here. You generate these holiday junctions. So holiday junction here, holiday junction right here. It's going to work as well. These, of course, are shown as X of 2 strands versus each other and continuous strands on the other side. Since DNA is double-stranded, wrapped around each other, these are actually completely symmetrical structures. And one of the things that can happen with these holiday junctions is they can move. And so this is the holiday junction branch migration process, and this is what usually leads to gene conversion. So it's not so much the repair process that happens right here. It's usually this branch migration. And so what happens here is you end up breaking a base pair, forming a base pair, breaking a base pair, forming a base pair. And then this can literally be thousands of nucleotides that these holiday junctions move before you have holiday junction resolution, which is cutting either this strand or this strand and religating them to each other. And in this process, you can either end up with patches where the ends of the chromosome are the same, or crossovers where the ends of the chromosomes are actually different. And so if you've had a gene conversion process, and often again this will be branch migration, what happens is you have different alleles. And so I always say I don't change my slides between my original one and my review. I lied because this is that change. So A allele, little a allele, big A allele. And A complement, that's what this little apostrophe here stands for. So what happens is you have a little a allele and the complement of a big A allele that are now next to each other, probably because of branch migration. Those alleles have different bases in them. And so that's where you have mismatches. The mismatch repair machinery comes in, sees a mismatch, makes nicks on either side of it, and repairs that mismatch. The problem is here is that now we don't have, like we had in replication, a nice indication, oh, this is an old strand, this is a new strand. Even in the case of replication in bacteria, methylation is going to be mixed here, and certainly in the case of meiotic recombination. There's no extra nicks on one strand versus the other. So randomly, you'll end up with one of them getting repaired and the other one not getting repaired. And so this is what can lead to that non-2-to-2 segregation process. So that's where the mismatch repair comes into gene.
Does this also happen through nucleotide excision repair?
In theory, it could, but what nucleotide excision repair does is it actually recognizes the structure that's not just a problem with the base pair itself. It'll be some other kind of problem with the DNA. Yeah, and so the mismatch repair machinery, the overall mechanism is the same, but how that is recognized as being something that needs to be repaired, that very first step is different between nucleotide excision repair and base excision repair. But yeah, but once you have that first step, once you make the nicks, everything else is the same. Okay, transposons. Maybe we'll get to mRNA modification at the end here. Really briefly, 3 major flavors of transposons, although somebody pointed out there are some other ones as well. But the major ones we're going to talk about are the DNA-only, the retroviral-like, and the non-retroviral retrotransposons. The DNA-only ones are very common in bacteria, although these are the first ones that were discovered, again, by Barbara McClintock. Ridiculously well-deserved Nobel Prize for that. How do you know, if you're just sequencing DNA, which of these kinds of transposons you have? DNA-only transposons have these short inverted repeats at either end, have a transposase gene that is encoded in the middle of them. Retroviral-like retrotransposons have directly repeated sequences at either end. Take virology, we'll tell you how they get them. And these have reverse transcriptase and integrase proteins in the middle of them. Those integrase proteins act a lot like transposase proteins. Non-retroviral retrotransposons have a reverse transcriptase encoded in them, no integrase, and these poly A tails. these two, retroviral-like and non-retroviral retrotransposons, move via RNA intermediates and DNA-only, just through DNA-only intermediates. Hopefully everybody's clear on, direct repeats and inverted repeats. Direct repeats, same sequence, same strand. Inverted repeats, same sequence, opposite strands. Good? Okay, so these DNA-only transposons, they will jump from one place in the genome to the other through the activity of the transposases. Transposases bind at either end of your transposon. They're monomers that bind to each other. They bind in opposite orientations, which is how you have those inverted repeat sequences at either end. They then chop this piece out, pick it up, take it to somewhere else in the genome, and in the process of going to somewhere else in the genome, it cuts that genome at an offset location. And so you end up with this overhang sequence, which is shown here in black. That's what gets repaired. What this means is if you're sequencing a genome, sequencing along, you end up with direct repeat, inverted repeat, inverted repeat, direct repeat. And just by seeing those sequences, you can say this was a DNA-only transposon. And you've seen how it's moved because of the direct repeats binds it to the transposase with those inverted repeats. Retrovirus integration actually happens in an extremely similar way. As I mentioned before, the integrase in retroviral retrotransposons is extremely similar to the transposase. It binds to, actually now, not inverted repeat sequences, but it binds to either end of the genome, and it's actually slightly longer than the piece that gets integrated. This then cuts the viral DNA, also cuts the target chromosome, which is not a specific sequence. It's usually a region of the genome which is being actively transcribed because it's got to get all of this stuff to the DNA. If it's completely compacted, they're not going to have integrations there. This is a problem if you have rapidly transcribed genes. But when you have this cut process, again, it's offset, gives you a little gap. That gap gets filled in. Because you have the fill-in of this gap structure, you're going to end up with direct repeats on either end outside your actual transposon and the direct repeats that you have because this is a retroviral-like retrotransposon. To see if it's a non-retroviral-like retrotransposon, it has that T stretch at the end, and the reason it's got those T's at the end because it was transcribed by your RNA polymerase II, and that's where it will cut and serve as the template for your reverse transcription. The rest of the reverse transcription happens in a different way. So now finally we get to talk about RNA, much more important and interesting. How do you make RNA polymerases? Again, you do not need a primer, which is one of the fabulous things about RNA polymerases. So DNA primase exactly the same way. Starts without a primer, lower fidelity than DNA polymerases, but is not something which is being transferred from generation to generation. One thing that I neglected to mention, I think the first time around, is that the number of bases that are actually paired in the RNA polymerase between the new RNA that's getting made and the template DNA, which is being transcribed from, is actually relatively short. This is actually quite a short, maybe on the order of 5 or 6 nucleotides. And this is important because you remember how does termination happen? Termination happens when you pull that RNA out of the RNA polymerase. So pulling it out, it's a lot easier to pull out if you don't have that many bases which are attached between your template and the RNA itself. Relatively slow, 50-odd nucleotides per second relative to the DNA polymerases, which are all faster than this. And as we'll see later, translation is the slowest of these processes. Great thing about RNA polymerases is they don't need primers. The downside of that is where do they start? And so that is the whole process of promoters. And so people seem to be rather confused about promoters. A promoter is literally where you have binding of the RNA polymerase to the DNA in order to say this is where you're going to start transcription. So That's the process. Once you have binding, then you have what's called this closed complex. Closed is just literally the two strands, which are DNA strands, are still attached to each other. Open complex formation is pulling those two strands apart. Helicase activity, TF2H in eukaryotic systems, the RNA polymerase itself in both bacteria and archaea. Once you have open complex formation, then it can start making nucleotides, and in many cases just spits them out. It doesn't start and go any further. Once this polymerase decides that it's going to continue to transcribe, there's a big change in the structure, a conformational change that happens in the RNA polymerase. It now becomes very processive. Here it was not processive at all because it falls off, starts again, falls off, starts again. This change leads to transcription, gets to termination, again, falls apart, and you have this whole process go again. So it's the binding of the promoter, which is the really critical aspect of things. And so a lot of people ask me about this particular slide. -10 and -35. How do you get -10 and -35? So -10, -35. There's also a +1 way the heck over here. So +1 is where transcription starts. And so transcription starts, in this case, going this direction toward me, out the classroom door here. And so that's where you start. That's your +1. If you count backwards 10 nucleotides, you find this conserved sequence in a bunch of promoters. You go another 25 nucleotides, and you get to this conserved sequence. And so it's these conserved sequences that the RNA polymerase is going to bind to, but also where it's actually starting. And so that +1 is also there. It's got to be there because that's where the polymerase is actually starting. And what that tells you is this RNA polymerase is a pretty darn big complex in order to get transcription to start at a particular place. And you've got lots of nucleotides to get you your specificity because you have to make sure they're going to bind to and start at the proper place. How some of these are used in some cases and not in others is a question that, again, we will address after the midterm. So just really briefly in terms of the RNA polymerases, bacteria, eukarya, and archaea are always similar. Archaea are more similar to eukaryotes. Eukaryotes, however, need all of these extra general transcription factors, TF2D, TF2B, E, and H. We care about D, B, and H as far as this course is concerned. D binds to the Tata box. B binds to the B recognition element. H has kinase activity and helicase activity. What does it bind to? B recognition element and Tata box, but also initiator elements and downstream promoter elements. And we saw this really amazing structure from Eva Nogales' lab. And actually, she's coming here. There's a microscopy convention happening in a couple months. She'll be here. Should be really awesome. Probably talk about this, actually, where we have interaction of TF2D sort of binds here and then kind of walks the Tata box over to here. And you get transcription to start. Archaea, nice simple system. Capping, we've got 2 more minutes to go here. We already talked about a 7-methylguanosine, 5' to 5', phosphotriester bond here. Guanyl transferase does that. Methyltransferase methylate this as a cap structure, which is then bound by the cap recognition complex, allows you to get export and, later as we'll see, translation. Splicing, 2' to 5' phosphodiester bonds through the activities of all of these snRNAs, which bind to the splice sites, branch points, etc. All of these RNAs together give you specificity. It's pulling these RNAs apart and putting them back together, which gives you your actual splicing that takes place. All the ATP hydrolysis here has to do with rearrangement of RNAs bound to the DNA. Probably the last thing that we'll talk about now is how you get splicing, how you decide. Mostly U1 is where you're going to start your intron. U2 is where you end your intron. What's really key here is where U2 is binding. So if U2 binds, you'll get splicing here. If it doesn't, you won't. And that'll be due to the interaction of these other proteins in the exons. Talked about that here. Really cool stuff that I did talk about. I reposted online. We'll spend some more time if you want to look at that, if you like, in extra office hours. And then we had poly A tails and export. And then finally, you modify your ribosomal RNA. Good luck on Friday. Bring your big scantrons. And if you have more questions on this, come find me.